is Joan Javiña, and I will be showing you the process of how muscle contraction takes place. I will be drawing the illustration. It would be a rough illustration of how it takes place, but in the span of this video, I will be flashing the real illustration of how it takes place. The first one would be, this would be represented as the axon branch. And this would be the representation of the sarcomela. And right here would be, this space right here would be the T tubules. So inside would be the sarcoplasmic reticulum. Just imagine that inside the sarcoplasmic reticulum, there would be your muscle fibers, your myofibrils, your actin myofilaments, and your other filaments. So this is the sarcomela. And this right here is the T tubules. And this is the sarcoplasmic reticulum. So let us start this process. The process starts within the axon branch. Inside the axon branch, there are the motor neurons. The motor neurons are the specialized nerve cells that stimulates the start of the muscle contraction. So without motor neurons, muscle contraction wouldn't be possible. So motor neurons stimulates what we call the action potential. So what the action potential does is it initiates the muscle contraction. So imagine the action potential traveling to the muscle fibers and reaching the presynaptic terminal. The presynaptic terminal are the neurojunction terminals that are just called the presynaptic terminals. In general term, it is called the synapse. So once the action potential reaches the presynaptic terminal, it will then meet a series of voltage-gated calcium channel. What this voltage-gated calcium channel does is that it releases calcium. After the calcium is released from the voltage-gated calcium channel, it will then stimulate the exocytosis of vesicles that holds the neurotransmitter. So the neurotransmitter inside this vesicle is called the ACH, or the acetylcholine.
In the left side of this illustration, it shows how the nerve impulse travels along the axon branch to the axon terminal, with the labeled parts of the synaptic vesicle containing the ACH or acetylcholine, the folded sarcolemma, the receptor sites bind with the ACH, and the synaptic cleft. While on the right side of this illustration, it shows the whole unit of muscle fiber with the labeled parts of the capillary, the axon branch, and the axon terminal.
Once the ACH or acetylcholine is released, it would then travel to the synaptic cleft and it would be binded into a receptor. So this receptor is a sodium channel. So basically what it does is it opens up the sodium channel gates and allows sodium or Na inside the muscle cells. So once the sodium successfully got through the muscle cells, it will then cause the skeletal muscle cell to depolarize. Once the depolarization takes place, it would stimulate a depolarization wave, and it will travel through the T tubules. The polarized wave will continue traveling down the T tubules, and then it will meet our second series of voltage-gated calcium channel. So this is our voltage-gated calcium channel. And this is our depolarized wave. Once the depolarized wave meets the calcium channel, it will then release calcium inside the sarcoplasmic reticulum. And inside this sarcoplasmic reticulum, we already have a calcium pump. So what it does is it releases more calcium with the help of the ATP, or what we call adenosine triphosphate. So what the ATP does is it powers the calcium pump. The calcium pump is the active transporter of the calcium. Our sarcoplasmic reticulum is already filled with calcium. What will happen next is the open voltage gated channel of calcium will then allow the release of calcium outside the sarcoplasmic reticulum. And when that happens, this is the stage where calcium is binded with the troponin. The important function of the troponin is that it has the binding sites of the calcium. So troponin is attached alongside the intervals of the actin myofilaments. So here is an illustration of how the troponin looks like. You can see the blue one would be the tropomyosin, and the red one is our troponin. So once troponin is binded with calcium, it would then move the tropomyosin, and our myosin would be revealed.
This is the illustration of our actin myofilament. The green line or wave is the tropomyosin. The blue circles are the actin strands. The red circles are the troponin. And below the chain is the two heads of the myosin molecule and its rod. Now that we're in the final stages of the muscle contraction, here is the illustration of an actin myofilament inside our sarcomere. In the first level of this illustration, in the thin filament, this is where the calcium is binded with the troponin. It will then move the tropomyosin so that the myosin site would be revealed. The myosin would then be binded to actin. When the myosin is binded to actin, that is when the cross bridge happens. In the stage of power stroke, after the myosin is already binded with the actin, it causes a shape change that then causes it to kick off the ATP plus P. In these final stages, now that the ADP plus P isn't there anymore, nothing will hold off the position of the myosin head, causing it to contract. It caused the whole sarcomere to shorten. And if the sarcomere have shortened, it is a conclusion that our muscle have already contracted.
Now, if ATP binds again, the myosin head will return to its cocked position, making it contract again. So that right there is the whole process of how a muscle contracts.